talking about how sexual orientation may have or may not have anything to do with epigenetic changes, and then talk about epigenetics and asthma. And if we get there today, I will show you the current grade distribution in the class and my sort of current thoughts about where letter grade cutoffs are, with a whole host of caveats, but we'll see if we get far. Before that, I was inspired by the question about whether or not Klinefelter syndrome patients, that is, individuals that are XXY, do X chromosome inactivation. It's a great question from last class. I promised I was going to go look it up, and here I am reporting back to you. I couldn't find any full articles that I had access to from Fresno State, so all I could find was the abstract. So what I did was I went to Google, basically, and I typed in Klinefelter X chromosome inactivation. And this article that was published in the American Journal of Medical Genetics popped up. And so at least I can see the sort of outline of what this manuscript would look like if I could get the full version. So you can see it on your tablet if you've downloaded the slides, but what do you think? What does that abstract tell us? Do Klinefelter syndrome patients do X chromosome inactivation or not? It's in there somewhere. Their hypothesis is that skewed X chromosome inactivation occurs. So they're asking, does X chromosome inactivation occur in Klinefelter syndrome patients? So do you see anything that suggests that X chromosome inactivation does occur?
They say maybe.
Where do they say maybe?
The last 3 sentences. They say that it does suggest that it's common.
Skewed X inactivation may be common. But now, this is a specific topic. There are 2 topics here. They're nested. There's a hierarchy. There's X chromosome inactivation, which is this process of epigenetic change happening only in females or in females plus Klinefelter syndrome individuals, where one X chromosome gets turned into heterochromatin and transcription shuts off. But beyond that, there's also this different topic, the skew. So we have X chromosome inactivation. They're just trying to ask, does it happen or not? Yes or no? And if yes, then the question that they're asking on top of that is X chromosome inactivation random or non-random? So here's an 8-cell embryo. You can't see the ones in the back because I'm trying to draw this as a sphere, so we're looking at the front 4. So if this is the stage at which X chromosome inactivation occurs, let's say there's one X chromosome, we can talk about a Klinefelter's patient. Let's say that this Klinefelter's patient is a Klinefelter cat, an XXY cat, and that one X chromosome has a gene that makes red coat color and one X chromosome has a gene. So that would be that X chromosome. The other X chromosome has a gene that creates a black coat color. So if at the 8-cell stage one of the X chromosomes gets turned off, and if that choice is random, so these are the 2 variables, in this embryo we're going to fill in each cell according to which X chromosome is still active. So it's either going to be red or it's going to be black. What would this look like if the process of choosing which X chromosome to turn off is random? How many of these cells that we can see, how many of the 4 would be red? 2. Half. So if this process is random, then the X chromosome that has the red producing gene would be turned off 50% of the time. And there would be little X chromosomes. Those are the inactive ones. And The active X chromosomes would be the red version of the X chromosome in the other half of the cells. So we'd have an embryo that's 50% red, 50% black. That's what happens if X chromosome inactivation is random. It's chance whether or not the black version or the red version of the X chromosome gets turned off. Okay? Pretty straightforward. What they're asking about in that abstract we were just looking at was non-random, or what they call skewed X chromosome inactivation. That would be a situation where you look at an embryo and it looks like this, for example. So by chance, this is random or non-random, I don't know. All of the cells have turned off one copy of the X chromosome, never the other. So in every one of these cells, the X that contains the red pigment-producing gene is active, and in every one of these cells, the other allele of the X chromosome has become inactive or turned off. That's skewed, also known as non-random inactivation. It's a non-random pattern. One chromosome gets turned off more than the other. But if you flip back to that abstract slide, I'm not going to now, they did show that there are data that suggest that X chromosome inactivation does occur in XXY individuals. If you've got two X chromosomes, even if you've got a Y chromosome also, you still undergo the process of random, sometimes, X chromosome inactivation. Any questions about this concept of random versus non-random X chromosome inactivation, skewing? If you've got 8 cells, 4 of them should turn off one X chromosome before the other. If you've got 16 cells, it should be 8 and 8, or approximately 8 and 8. I suppose we should do some chi-square analyses, but maybe not now. And the reason that was such a great question to have asked about Klinefelter syndrome and that X inactivation is because it leads us right into this slide, which I ended last class saying we were going to talk about today. So here it is, getting back to the concept of whether or not epigenetic change might have an effect on sexual preference. So what was the hypothesis that we looked at last class about? What was the study? Talked about epigenetics and sexual identity for twins. Spit in a cup, I'll tell you what your sexual preference is. So what were some of the conclusions we reached last class about that study?
It wasn't a very sound study.
That's the big conclusion. There are some issues with it. So what are those issues?
You're saying that the DNA in the saliva was different than DNA in other places?
Okay, so it could be that, yeah, is it appropriate to ask if the DNA in saliva has certain epigenetic marks, because that might not be representative of epigenetic marks elsewhere in the body that might be more important in determining sexual preference. Sample size. Why a small number of twins? There was also a bias in gender. They looked at male twins, never female twins. Not sure why. That should raise a red flag. A question in your mind, please. Why did they do that? This is a separate study. And this has some flaws too, and I'd like you to think about this as I'm yammering on up here. See if you can pick up on things that you question, you wonder about. Is this true? So they had the exact same question we just discussed about skewed X chromosome inactivation. Is it random or is it non-random? And the hypothesis here is that the mothers of homosexual sons have non-random or skewed X chromosome inactivation. That says it in the title of this up here on the screen. Extreme skewing of X chromosome inactivation in mothers of homosexual men. So they do exactly what I just drew on the board. They took cells from mothers of homosexual men, and they asked, in all of the cells we take from a single mother, did we get a 50-50 division? Half of the cells turned off one X chromosome, half the other, or is it non-random? So here are the control mothers. What would a control mother be in this study? Right, no sons that are reporting to be homosexual. So mothers that have all heterosexual sons. Anybody have any questions, by the way, at this point? Concerns with this study? We haven't talked about the conclusions of the data yet. Go ahead.
Would the control mothers also have a difference in X chromosome activation?
So that's exactly why these are the control mothers. We want to know, is there a difference between mothers that only have heterosexual sons and mothers that have homosexual sons? That's why this is the control group. So we're going to compare the data up here in panel A to all of the other panels. Yeah.
Kind of lens for all of them, like how many sons do they have, and what is each? How do they? So even if it was like one homosexual son, if they have 5 heterosexual sons, could that?
So that's a great question. Yeah, absolutely. We want to know details, and you should be asking things like, how many sons? Was there a minimum requirement? Like, we only include mothers in this study who have a certain number or more of sons. Because we know now already in this class, by chance, things can go wrong or different than you hypothesize, not wrong, if you have low numbers of individuals that you're studying. So if a mom has one son, and that son is gay, it's 100%, that skews the data. So maybe the authors, and maybe you should look up the paper and find out exactly in the materials and methods section of this manuscript, what did they do to include mothers in the study? Was there a minimum number of sons they had to have for them to be included in the study? Or do we have some data up here that are from mothers that had one son, or worse, mothers that had 0 sons? I mean, I'm hoping that there are no mothers up here that didn't have any sons, because why would they be in the study? But it's important for us to ask these questions, because you never know what those scientists did, unless you go look at the details. Okay, so axes. Pretty straightforward. N is number of sons, number of mothers in this first panel. And the x-axis looks like a percentile range. So what percent of cells had one X chromosome turned off? And they define down here in the figure, which you can't see, you'll see it on your screens, but not mine. They define what extreme skewing means. Down here, greater than 90%. So any mother they find more than 90% of her cells have one of the 2 X chromosomes turned off, they call extreme skewing. And that's all of these columns that are filled in black. Yeah.
Another question about it is doesn't mention daughters either.
Right, so once again we have that sex bias. We're looking at sons, not daughters, for some reason. So what percent of all of the mothers had extreme X chromosome skewing in the control group? That's that fraction I wrote up there. 4 out of 103. So if you sum up, right, there's 6 mothers that had 50 to 55%. And that's what, maybe 12 there, and 14 there, and 15 there. So if you sum all that up, they looked at 103 control moms. Only 4 of them had this extreme skewing, where most of their X chromosomes turned off one copy of the X, not the other. How does that differ from the rest of these graphs? So look at the bottom right one, for example, down here, panel D. Mothers of 2 or more gay sons. What happens to those mothers? Yeah, now we're up to something like 25% of mothers that have 2 or more gay sons have this significant X chromosome inactivation skewing, where somehow there's this correlation, is what these authors have determined. That if a mother has multiple gay sons, she's more likely to have only one of her X chromosomes turned off throughout most of her body, not randomly. So if I was a better artist, I guess I can do this stick figure style. Let's see, dress. Hey, there's a mother. Okay, multiple gay sons. And then we've got a mother that has, she's wearing a dress, obviously, not putting shoes on her. 0 gay sons. So if we use this red versus black coloring, how would I draw the mother on the left? So a mother that has multiple homosexual sons. She'd be mostly one color. Most of her cells have turned off one X chromosome, and a few of them have turned off the other. That's opposed to what happens, according to these authors and their data, in the control mothers, where they see mostly random X chromosome inactivation. Half of the cells have one X turned off, half the other. So correlation. Mom's and X chromosome inactivation, random versus non-random, and mom's, what's the sexual orientation of your sons?
Well, I would assume that testosterone came from their father. I don't know, it's just an assumption. Would there be any correlation with levels of testosterone?
So we have lots of questions. And of course, since we don't know really the basis of homosexuality, as far as I know, then it's hard to come up with hypotheses. So yeah, it could have something to do with testosterone levels. We could measure that. It's been done. But this one just finds the specific correlation between X chromosome inactivation in the mom and sexual preference of her son.
Where did they get the cells? So is it like from the ovaries where like that genetic code is different than maybe a somatic cell?
So sorry, say that again?
So for the inactivation is, because we're talking about like epigenetics and everything too, are they just trying to, their genetic makeup could cause the same level of inactivation in the sun?
Well, so we're not talking about X chromosome inactivation in the sons. We're asking about the mother. And this is that process where, so early in the mother's life, before she looked like this, she looked like a single cell, and then 2 cells, and then 4 cells, and then many divisions later, you get billions of cells, right? So something happens early in development, right? This is review from last class, where at some point during early development, small number of cell stage, each of the cells will randomly turn off or non-randomly turn off one of the 2 X chromosomes. So this is getting at a big question that's a really important question, so I'm glad you asked that. So what this suggests is that if you don't have random X chromosome inactivation, this correlation, we're not at causation yet. We don't know that one means the other, we just know that there's this correlation. If you happen to be a mom that has non-random X chromosome inactivation, there's this correlation with having homosexual sons. Does that mean that it causes, or is just a correlation? We don't know yet. The big issue with this study. What are some other concerns and questions? There's a really big issue here. Beyond all the ones we've talked about already, the previous study, the saliva, spitting and cup, and all that stuff.
I don't think it's the question you're asking for, but I would think demographic size, or where they found, where they picked up these moms, whether it be
Yes. What was the distribution of the participants in the study? So was it all from the same ethnic group? Was it all from the same geographical location? Was it all, was this a random sample of women, or was it non-random? That's a good question to ask. You should all be thinking this question right now in your heads. So blurt it out. What are you thinking right now? Why the hell are you staring at me? What do you wonder about this study? Let's have a frank and honest and open discussion.
Nature versus nurture?
Okay, so is it genetics or is it environment?
Well, why didn't we look at guys?
Why didn't we look at guys? Well, we looked at sons, but not daughters. So why did we only look at guys and not? But why didn't we look at fathers? Okay, fathers don't have X chromosome inactivation, but this is all circling around a really important, key, fundamental question to this study. Yeah.
So, does that mean sexuality is related to your gender? Like, are those together?
So, does something about sex chromosomes have something to do with your sexual identity?
Yes.
Okay, now we're drilling down, we're getting closer to this really fundamental, that's the perfect question to ask. It's getting down to this really fundamental question. What I want you to tell me is, what's the hypothesis of this study? If this is real science, it is, it's a scientific study, it was published. If this is real science, there should be a hypothesis. So what's the hypothesis? Right now we've got a correlation. Non-random X chromosome inactivation, more gay sons. What's the hypothesis? Tell me a story that explains how this works.
The X inactivation affects your sexuality.
Right, so we start out with this sort of vague hypothesis that, yeah, somehow, it's the question, how does X chromosome inactivation in the mom lead to sexual preference of the sons? What does your mom's X chromosomes have anything to do with the outcome? What's the connection between on the top and on the bottom of this slide? What's happening?
I don't know if this is what you're looking for, but it makes me think of the calico cat where it has to have one of the black and one of the orange, so say like one gay, one straight X gene. So that means that some moms ideally should have all gay sons, and they're not showing that in the study.
Oh, so we don't know the percentage of gay sons from the mom, so maybe but we know for sure, we mentioned this at the start of class last time, there is not a single gene that controls sexual preference. It's not on the X chromosome. So the main issue with this study is that there isn't a plausible explanation for why random X chromosome or non-random X chromosome inactivation would affect sexual preference. That is, there's no simple story that can be told that explains the link from the top to the bottom. Why does your mom's X chromosome inactivation pattern have anything, why would it have anything to do with sexual preference?
So they'd have to do another study,
They better.
figuring out what the effects are of being skewed or not.
Right. So a good follow-up to this study might be you take a bunch of women that have sons and you don't look at their X chromosome inactivation first. You ask how many gay sons do you have, you sort them into two groups. So you hypothesize now, this is an initial preliminary finding. We've looked at X chromosome inactivation, we saw a correlation with whether or not your sons have a homosexual or heterosexual preference. You do the reverse. You find a bunch of women that have a lot, have a lot of heterosexual or homosexual sons, and then you do it backwards. Then you look at their X chromosome inactivation pattern and see if you can predict which moms have skewed X inactivation based on the sexual preference of their sons. And that's often how these sorts of studies get validated. You run the study one direction, looking at correlation in one direction first, and then you reverse it and see if it's still true.
Couldn't even that be falsified? I mean, because there's that whole idea of the Kinsey scale, you know, so like, how do you know if you truly have one or the other? It's not necessarily black and white.
And then, yes, black and white is useful description, non-specific. So absolutely, both the study we talked about last class and this study suffer from this main concern that this is all based on self-reporting of sexual preference by sons to their mothers. Who knows whether or not that's accurate in the first place, right? Son's in the closet, hasn't told his mom yet, so she doesn't know. So doing these sorts of genetic studies, every time you see a study like this in the media, it's good to start asking, think really, if you're going to try to come to a conclusion yourself about whether or not you believe it, believe the conclusions, to dig into some of these details first. Okay. I went for a long run this morning, as I often do. And I got home, I came to work, and my wife texted me. My daughters put up the flag at their elementary school, and they put up the US flag, plus they put up an air quality flag. I don't know if you guys know about this, but my wife texts me and says, orange air quality today, don't go outside and breathe. I said, great, thanks. I spent like an hour and a half running this morning outside, and now I learned that I'm not supposed to breathe. The reason that that's relevant to me, well, I don't have asthma, my wife does, is of course, you're probably all aware that we have really shitty air quality in the Central Valley. And every year, I didn't update this for this year, but every year the American Lung Association puts out this big report, State of the Air, and they rank metropolitan areas, and of course, California consistently ranks at the top of the worst air quality in the nation in terms of particulate pollutants. So there you go. 2015 and 2016, Long Beach was the worst, but look, we're down there at number 4. We're holding strong at number 4. But we're surrounded by every other place in the Central Valley, Bakersfield, Modesto, and so forth. Right? So that's US rank. So Central Valley and Southern California is awful for air quality. And that's true both for, so these next few slides I'm going to skip right over. Basically, it's true for different types of pollutants. So this is short-term particle pollution. The next slide is year-round particle pollution. Bakersfield's on top. But, we took number 1 in 2015, but we lost it to Bakersfield 2 years ago, last year. Ah. And in particular, where we live, the reason that this air quality study focuses on the Central Valley is because we have a lot of at-risk individuals. We have, right, Fresno is the 5th largest city in the state, or is it 4th now? So we have a lot of people here, we have a lot of elderly people here, a lot of infants, a lot of young children, a lot of people that are immune compromised, and so forth. So there's a huge population of people that are at risk for suffering from this poor air quality. And there are lots of pollution sources. Great. Now, in 2010, this study was published. This is from a pediatrician who works at Stanford, so Palo Alto, Bay Area. And she also has a practice here in the Central Valley. So she teaches at Stanford, she works here, so she goes back and forth. And she had noticed that we're all Californians, that doesn't mean much in the context of this study, but she travels just that far, a couple hundred miles, Bay Area to here and back, sees huge differences in the number of children that are affected by asthma. So of course she starts thinking, well, is it nature, is it nurture? Is it just exposure to bad air quality in the Central Valley that makes a lot of children, and adults too, have problems in the Central Valley? Fortunately, there's something that's known about how asthma is produced. So briefly, this is a chart of different cells. Now we're talking cell biology for a second, not genetics. We'll get back to genetics in a minute. Different cells that are involved in our bodies in the immune response to these pollutants. The key player here are T helper 2 cells, or Th2 cells. If you have too many of those immune cells in your body, those produce the effects the symptoms of asthma. Wheezing, shortness of breath, constriction of the air tubes, and so forth. But look, there's arrows and there's blunt arrows up here. So what happens is you get antigen-presenting cells that bind to, for example, there's allergens, are detected by antigen-presenting cells. Those cells turn on, right, solid arrow, or arrowhead, they turn on T helper 0 cells. And those cells activate T helper 2 cells. So we have this direct link at the cellular level between bad air quality, what you're breathing in, and the activation of the cells and the development of cells that cause asthma and asthma-related symptoms. Based on this plot up here, the interactions between these cells, what would help people that have asthma? What cells would you want to have around to control asthma? Which ones? Tregs, Tregs. T cells that are regulatory T cells. These ones here. Because they do this. They repress the development of T helper 2 cells. So you want to have Tregs to prevent your T helper 2 cells from going crazy and being overactive and producing asthma-related symptoms. And that's the hypothesis down at the bottom. So Dr. Nadeau hypothesized that maybe what was happening in the Central Valley was that people with asthma, didn't have enough T regulatory cells to repress the T helper 2 cells. Now we get back to genetics, just really briefly. This gene here, FOXP3, is important in producing Tregs. So when this gene is active, that gene causes the production of T regulatory cells. So ultimately, the genetic hypothesis was, down here at the very bottom, that the FOXP3 gene needs to be turned on, you produce Tregs, that represses the T helper cells, no asthma. So if you don't have a FOXP3 active, if that gene's not being transcribed and not being translated, then you don't get Tregs, and you're more likely to get asthma. That's the hypothesis. So we set up the battle between the trees and the bulldogs. Hopefully the bulldogs win this one. So what Dr. Nadeau and her colleagues did was they measured two things. This one, they look at T regulatory cell function. So how well do those T regulatory cells repress the Th2 cells? If your Tregs are functioning at 100% up here at the top, would that correlate with having asthma or not having asthma? Not. So if your Tregs are working, that's suppressing the Th2 cells, you should not have asthma. So what we see up here are data points from the SNA group. That's Stanford S, NA, no asthma. So these are kids from Palo Alto that don't have asthma. Each one of those boxes is a single data point for a single kid. So in general, one kid, in general, if you live in the Bay Area where the air quality is good and you don't have asthma, your Tregs are functioning pretty well. What happens if you're in Stanford and you have asthma? That's the SAA group. Still, so how are your Tregs functioning? Pretty well, 90 to 60% function. Okay, so that's Stanford, asthma versus no asthma. What happens when you move into the Central Valley? Even if you don't have asthma, your Tregs are only functioning at like the 70% function. And if you're the people that have asthma in the Central Valley, then you get down to your Tregs are functioning as poorly as 30% of their maximal possible function. So this suggests that somehow, if you live in the Central Valley, something's going wrong with your T helper cells and T regulatory cells. Well, it's entirely environment. So this is, what we're getting to is the impact of environment on gene expression. How our poor environment affects the production of the gene, the FOX gene, that's important in turning on the T regulatory cells. So I'm going to ask you to make a prediction. I'm going to ask you to draw something on the next slide and send it to me via Google Classroom. We'll talk about this at the start of next class. So we know these things, I'll let you read them. I don't need to read off the slide. But the critical part is that third bullet point. When Dr. Nadeau published this study, or when she started this study, it was already known in mice that environmental factors can epigenetically modify that gene, the FOXP3 gene. So what we're getting at is, does asthma somehow change the expression of the FOXP3 gene from on to off? So you make the prediction. And you don't have to do this right now, but before next class. Attach this to the Google Classroom. I want you to make a prediction. Is this gene going to be more heavily methylated in the Palo Alto kids or the Fresno kids? And is it going to be more heavily methylated in children with asthma or without asthma? And after you do that, or as you're doing that, draw in this chart for those 4 different types, right? We had Fresno asthma, Fresno no asthma, Stanford with asthma, Stanford no asthma. So on the x-axis, instead of Treg cell function, I want you to plot what? Yeah, FOXP3 methylation. So I want you to draw, please, what are the data going to look like if you did this experiment? So how would you predict FOXP3 methylation would change among these 4 different groups? That's it. So draw on this slide, take a screenshot, attach it to the Google Classroom assignment I'm going to send out, and then send that to me by next class. The other thing to do for next class, you can start now if you want, but actually don't start now. For next class, also, work on this quiz. I've placed a link. This is different than explain everything. This is different than Socrative. This is a Qualtrics, an online web-based multiple choice, hit a radio button, move to the next page sort of survey. But please do that by next class as well. So those are the 2 things to do for next class. The prediction on the previous slide, this quiz, I think it's 9 questions, not too long. Lastly, since we've got 5 minutes, any questions at this point? Because after this, I'm going to move on to showing you the current grade distributions in the class. It's like a survey that's a quiz. It's a quiz. Okay. Now, showing you this grade distribution comes with a number of caveats, so I want to make sure, maybe I can't show you. I want to make sure that you all know this. So if there's a lot of talking, I'm not going to talk. And who knows? Maybe I can't show